Thanks for welcoming me. I'm a relatively new member of this Alexander community, and I'm still learning, but it's exciting to be here. And we've heard a lot of really interesting talks this morning that ASOs have managed to slip their way into a bit. So I am going to talk about ASOs, antisense oligonucleotides, which are the focus of Ionis. It's all we do as a company. And try to give you an idea of why we think this would be a good strategy in Alexander and why we're so excited about the data that we have so far. I will give a bit of a spoiler alert. We're not going to go into clinical trials tomorrow. We are hoping it will come. We are working toward clinical trials. You'll see some of the data today that we're excited about, but this is a process that you need to work through. We have a plan over the next few years. If that plan all goes according to schedule, we would expect to be able to start human testing. And how long it takes once you get into that to proving safety and efficacy varies. In this disease where it is so small, it would be a limited number of clinical trials. I think it would be a pretty efficient process once we get to that point, so maybe a few years after that. But that's generally the horizon. We don't have a lot of details on what those trials will look like. When we do, I swear we will share them with you, but I don't want to tell you today what it might be and have it not be that. So what I can commit to is that as a company, we're very excited about this. We have a long-term, lovely collaboration with Albie and his lab that's generating really nice data. We're forming a new collaboration with Amy Waldman and CHOP to gain their expertise. So as details come out about what's coming next, we will share that with you. Maybe through Albie, since he's the center of everything. All right. So, Ionis Pharmaceuticals, until about 2 years ago, ISIS Pharmaceuticals. I'll let you guess why we changed our name. We were founded, though, in 1989 with this sole purpose. We wanted to turn antisense oligonucleotides into a therapy. It's a brand new kind of drug, and I'll get into that in the next few slides. We were committed to that in a way that might seem stubborn, really, in retrospect. A lot of other companies tried it and said it'll never work. It got out. It's now working. We've had some success, and a whole new wave of companies are coming in. We're in Carlsbad, California, and we're relatively small by pharma standards. We're about 450 employees, maybe half of those in research. We do still have a lot of people dedicated to understanding ASOs. So there's this core research group that doesn't work on any particular disease. They just work on understanding the mechanisms of ASOs to better the work that we all do in disease-specific areas. So there are now 4 different drug discovery platforms, and I hope this will help explain what ASOs are and what they aren't. Small molecules, proteins, nucleic acids, which is what ASOs are in, and gene therapy. So small molecules have been around forever. At Neanderthal burial sites, there are herbal supplements have been found, or herbal stimulants. So we know, like, from the beginning of time, people have used chemicals that they found in nature to have pharmacological effects. Morphine was extracted from opium in the 1800s. That's a drug that's still around. Bayer aspirin came out, I think, in the late 1800s. So over time, we've gotten really good at extracting or creating chemical-based drugs. And now on the market, probably 85 or 90% of drugs are what we call small molecules. Proteins are a different kind of drug, also called biologics. So in the biotech era of the 70s and the 80s, we got good at using cells to produce drugs. So these are a little bit more difficult to make than small molecules, but they're a different kind of drug. Insulin, human growth hormone were 2 of the first. But now there are a number of really good ones. Any drug you see advertised on TV that ends in MAB, that's a monoclonal antibody, and that's a protein-based biologic drug. It was produced using cells to make the drug for us. And again, maybe 10 or 15% of drugs are biologics. I don't even think we rate 1%. We're so small. So nucleic acid drugs are pretty new. We've been working on it for 30 years. It took a long time to turn them into good drugs, to turn them into molecules that had good drug-like properties. So the scientific breakthrough comes early. In the 70s, we knew this is a technology that could work, but it took decades of really smart medicinal chemists to make it a good drug. And then gene therapy is something completely different. It often gets confused with ASOs. I'm going to put it off for now, but I'll circle back later to how we are not gene therapy. So you need to understand a little bit of biology for ASOs, this central dogma. And it's come up this morning, I'm sure most of you are well versed in it, but DNA makes RNA, makes protein. So proteins in your body are vital. These are These are molecules that they are the structural basis of cells. They serve chemical reactions, thousands of chemical reactions every day. They help protect you against invaders. They transport molecules. They store molecules. Proteins are essential. And DNA is it's an overused analogy, but it's like the recipe book. It's the instructions that live within your cells that tell your body how to produce every protein that you need. DNA is this double-stranded green molecule here, and what happens when your body needs to make a protein is that it unwinds a little bit to expose the recipe for the protein of interest. So generally it stays coiled up and nice and secluded in the nucleus, but when you need to make a protein, it'll expose that little region that's the recipe just for the protein you need, and a copy is made called RNA. That copy then can move out, and it finds its way over to a little machine that makes a protein. That happens again and again and again. So DNA turns into RNA, turns into protein. There are upregulators and downregulators in your body to make the kinds of proteins that you need. But this is a really well-established process and vital to life. You can interrupt that with an ASO. So an ASO is a little string of nucleotides. It kind of looks like DNA, it kind of looks like RNA. And if you design it so that it binds specifically to an RNA of interest, in this case GFAP RNA, it will bind to it and it'll break up the RNA, which frees the ASO to go find another RNA and bind to it and break it up. So we've interrupted that central dogma. With less RNA available to get to the machine, less protein is made. So it's really simple, and it was really cool in the 70s when somebody suggested this as a mechanism for a drug. It's just taken us a little while to get there. One of the cool things about ASOs is that you can choose the magnitude of effect. So if we put a little bit of ASO in, we'll get a little bit of destruction of RNA, and the bulk of the protein will still be made. If we put a lot of ASO in, we shift the balance of those arrows. Truth be told, we can't knock out protein production completely. Invariably, some of those RNA molecules will make it to the machine and a protein will get made. That's generally okay. We're generally not looking to completely eliminate protein. But at least what we're seeing in the mice, if we give a large dose of ASO, we could get 85 or 90% of the protein to be knocked out. And as we look forward to potential treatments for diseases like Alexander, we think that's probably sufficient. Okay, circling back to gene therapy for just a bit. If you look at this process, you could think, well, I've got a protein that's a problem. I could make less of it by attacking the RNA. But what about going back to the DNA and actually changing the DNA so that the protein that's made is not bad or is gone? That would be gene therapy, actually manipulating the DNA itself in some way. Introducing a new recipe to the recipe book or using molecular scissors to change the DNA itself. The latter in particular is nowhere near ready for clinical testing yet. It's another really cool development in molecular biology that over the decades will potentially turn into good treatments, but that's a long way away. So ASOs are not gene therapy. Also think about it, we're not changing the DNA with ASOs, so the RNA is going to keep getting made, which means what we're talking about are treatments, not cures. We're talking about treating these diseases like any chronic condition that you have where you take a medicine and it addresses the problem, but it's not a one-time cure. This slide's kind of gross, so I won't stay on it too long, but essentially it's saying that we can work anywhere in the body. So the ASO mechanism that we'll talk about, that I just talked you through, is one of many different ways that ASOs can work. We can actually upregulate proteins or downregulate proteins, depending on how we design that little molecule. And if you think about it, anywhere that you have a problem in the body because of a bad acting protein, you might address it with an ASO. So we've done work in all these areas, but what we're talking about today is the brain. And one element of using ASOs in neurological conditions is that ASOs don't cross the blood-brain barrier, which means we need to deliver them directly into the CNS. So in the programs that we've worked in, and in this Alexander program, we predict that we will need to use intrathecal dosing, which is an injection at the base of the spine. So we've talked about CSF, cerebrospinal fluid, which bathes the brain. Let's see if I can get this to work. And it also runs down the center of the spine. So the injection is below the spinal cord. So you're not at risk of touching the spinal cord with the needle, but it goes into this sort of sac down at the base of the spine. And because of the pulsatile mixing, which has been discussed today as well, the way the CSF moves around, we know from animal imaging studies that the ASO really does go nicely and distribute around the brain. So we're talking about bolus intrathecal injections. We have a lot of experience with this. About 5,000 patients to date have been receiving intrathecal injections. Most of those in the commercial space with Spinraza. So spinal muscular atrophy was our first success in neuro. I guess it's been about a year and a half since it was approved, maybe 2 years. But this is a really devastating pediatric neurological condition. It's actually the leading genetic killer of babies. And this was an ASO designed to upregulate a protein. So it was a situation where they didn't have enough. Here's a little picture of the box of it. So most of that 5,000 is patients who've been receiving the drug commercially. We also have clinical development stage projects in ALS, Huntington's, and Alzheimer's. So another couple hundred adults here who have received experimental investigational ASOs that might ultimately prove to be therapies for those conditions as well. So the big questions we have when we set out to design an ASO for a program on the clinical side, meaning in cells and animals, can we design an ASO that potently and specifically reduces GFAP RNA and therefore the protein? Meaning we don't want to hit any other RNAs. We don't want to accidentally knock it down anything else. We want to specifically just knock down that one protein. Do animal models of the Alexander disease condition benefit from treatment with ASOs? And can we establish safety in animals that would allow us to be comfortable going into humans? Oh, and to do this, we work in a number of different models. So you've heard about cells and mice, and Tracy told us about some exciting new studies with the rat. We do work in monkeys as well. These aren't transgenic animals, so this isn't that situation where you make them look like humans and you give them the disease. But in terms of getting through the required toxicology studies to get to a human study, we do work in monkeys because anatomically they're a lot more like humans than anything else on that chart. So what do the data look like? I'm going to show a little bit of data from Tracy's recent paper with the mouse model of Alexander. This is where Barrett originally had this invitation because my colleague who's in research, I'm in the development side, worked with Tracy on this project and somehow didn't manage to realize that she would be 36 weeks pregnant at this point, so I'm a last minute stand-in. But just a bit of that data. So as a reminder, DNA to RNA to protein, which if it's a bad acting protein causes disease. So the hypothesis is if we add an ASO, we would expect the RNA to go down, we would expect the protein to go down, and we would expect the disease to improve. And looking just at that first step, again, some data from Tracy's paper. These are 6 different brain regions. And on the vertical axis, we have GFAP transcripts, so that's how much RNA was measured in those brain regions. 2 different types of mice. The black circles are normal or wild type, meaning type found in the wild mice. And the red squares are the mice that are genetically designed to have a form of Alexander's disease. So for each of these plots, these are, again, just different brain regions, we have 2 different types of mice who receive saline and 2 different types of mice that receive ASO. And the interesting part when you look at the saline pairs on each of these plots is that the Alexander disease mice have a lot more GFAP RNA, which is what we would expect. We've mutated the GFAP protein to act like it does in humans, and in humans, you upregulate the amount of GFAP. So wild-type mice have low levels, GFAP-type mice have high levels when you just give them saline. When you administer an ASO designed to specifically target GFAP RNA, it's knocked out. There's no more GFAP. This is basically a not measurable level. This is approaching 100%. And when we look at the next step, so RNA has dropped. Is it true that protein has also dropped? Because that would make sense based on how we know the dogma works. And here we see that it does. We're just showing 2 regions, hippocampus and the cortex. Again, the black dots are the wild-type mice. The red squares are the GFAP mice. And you can see the effect of being a model mouse. But when you give the ASO, the protein is pretty much gone. And this is soluble or sort of free-floating protein, and then a couple different ways to look at the clumps. Another way to look at that protein is to take slices of the brain, and honestly, I don't remember which part of the brain this was, but here you've seen earlier today blue staining, meaning those are the nuclei, and these pink or fuchsia clumps are the Rosenthal fibers. So wild-type mice don't have any fibers. Alexander disease mice do. And when we treat them with saline, we see that we still have those pink or fuchsia clumps. When we treat them with the ASO, no more Rosenthal fibers. It's doing exactly what we would want it to do, which biologically makes sense. It's a pretty simple process. We know exactly the target. As long as the drug is getting to where it needs to be, it's doing what it was designed to do. But the big question is, does it affect disease? And that's a little harder to tell because of what we've discussed earlier, that these are mice, they're not people. And the mouse model in particular doesn't have a lot of the phenotype or a lot of the symptomatology that we see in people. So when we look at what Tracy was able to show in the paper, these top 5 lines in black font are disease symptoms that she sees in mice. So stress and immune markers increase, we're triggering extra GFAP, we've got a problem removing the trash. These sort of biochemical and cellular components of the disease are all improved with ASO. But what we don't have, and what's been discussed earlier with this model, is we don't have a real physical presentation of Alexander. So we don't know if we're helping cognitive dysfunction because this particular model just doesn't have it. Motor function, myelin changes, we just can't look at them in these models. So there's been a lot of talk about how valuable models are. I really like the saying, and I can't remember who said it, that all models are wrong, but some are useful. And here, we've fixed the things that we can fix, but how well does the model look like humans? We don't know. So we're really excited to work in the rats next, which show more of a phenotype, and see if we can answer this question a little better. When we lower RNA, because we know we can, which lowers the protein, because we know it can, does that help? Because that's what we need to know to go into human testing. So next steps, we'll continue the research collaboration that we've had with Albie and Tracy for a number of years. It's been a lovely collaboration and I think enjoyable on all parties. We'll work to establish the safety to allow clinical trials to be teed up and ready to go. We're starting collaborations with physicians so we can better understand the disease and better understand the history, like the natural course of the disease, because that will help us to design clinical trials. And that's where the relationship with Amy and her team comes in. And we'll also work on the side to liaise with regulators, go to FDA and talk to them about the disease so they can start to get familiar with it, talk to them about what our drug might do and how we might design a trial to demonstrate that it's safe and effective. So that when it comes time to do it, everybody's on the same page. Here's the trial we're going to do. If the results are positive, regulators will be receptive to that. Long list of acknowledgements. First and foremost, patients and families. I think maybe you don't appreciate how important it is to us to hear from you. I hope you do, because it is, and that's one of the reasons I'm here, is that I'm not an expert on Alexander disease, I'm a drug developer. And I can go to Amy and her colleagues and build that network, but I learn a lot from just meeting families and meeting patients and talking to all of you. And the contributions you make by participating in natural history studies, whether it's just a survey or contributing the story of your journey, is really valuable. It really does help guide us in what we decide to do. We also have, of course, the collaboration with Albie and his team. And I've highlighted in purple here the folks at Ionis who worked closely with Tracy on the work that was presented here, the data. But there is a much broader team at Ionis that is preparing for those clinical trials. So in addition to demonstrating sort of the proof of concept in animal models, we have a lot of work to do outside that, sort of not as exciting work, but just the preparations for getting ready for a clinical trial. So there's a pretty big team at our company that is dedicated to this. And I'll take back to them what I've learned from all of you here. And it'll motivate us. And then, of course, the CHOP collaboration is really exciting, and Amy's going to talk about that next, I think. Oh, and one more thing. Christina Boyer is at Iona, she's a colleague, and she's in what's called patient advocacy. So it is her role to make sure that we're incorporating your experience into our work. So if you want to reach out to her, I'm sure she'd love to hear from you. Even just stories, your patient journey, how you came to a diagnosis, first symptomatology, those kinds of things are really helpful to hear. So I put her, with her permission, I put her email address down here. If you want to get to me, I'm just A. Smith with the same little suffix. All right, any questions? And I have to warn you, I don't hear very well, so speak up.
When you developed the ASO for spinal muscular atrophy, could you just briefly talk about how you got the information needed for the FDA in terms of that the drug was safe? And then added to that is, what are some of the side effects that you've seen maybe in SMA?
Yeah, so I didn't work on SMA, so I'm not well versed in the details of that process. I know that team did some work in humans before having a lot of interactions with the regulators about what an acceptable package would be for a drug approval. And that's because it was a first, right? It was the first IT drug that we were developing. We went through a little bit more rigorous of a process, some small short studies first, a little bit of dose escalation, and then some longer studies. So that path was a little bit more extensive than what I think we can do now, because we have that experience under our belt, and we have those relationships with FDA where they were really happy with that development, and now they understand ASOs, so they're supportive. Safety, there are components of this that introduce risk. The biggest is the intrathecal injection. It's generally a safe procedure. In adults, at least, I don't know that we had it reported in the babies because they were babies, but there's a risk of post-lumbar puncture headache, which could be because you've taken off some CSF, or it could be because you have a small leak. That's really the most commonly reported side effect of the procedure. There are, of course, risks of worse things, but they are very rare. Like you can get an infection because you've broken through the skin. But those kinds of things are generally rare. And intrathecal injections are used commonly in chemotherapy treatments. There are spasticity treatments that are delivered this way. So it's not like plowing new ground. We know a lot about intrathecal delivery.
The fact of just introducing the ASO into the body, the central nervous system, anything that you side effects or unintended consequences from that it's significant or no?
There really weren't in SMA. So you can find the package insert. I don't know if I read them because I'm a drug development geek, but you can go online and find the paperwork that, when you buy an Advil, you have this little crumpled up, folded piece of paper in the box. You can read those and it will describe the types of side effects. I'm not familiar enough with that label to know what was in there. And we've had an evolution over 30 years. The first ASOs that we tried to develop were really pretty toxic. I mean, they were not well tolerated. First generation ASOs, I shouldn't say that, nothing that's on the market, but when you first attempted this, it was not well tolerated. And we've done a lot with medicinal chemistry to improve that. That's been part of the journey. Once we got to SMA, I think it was a pretty well tolerated molecule. Yeah.
So with the other diseases, were they mimicked like Alexander disease when you guys were testing them in like mouse models and rat models, or were they actually the actual disease that you guys were able to put into them to treat them, I guess? Or ours is more mimicked than the actual disease in them?
So is the question, how good were the models for these other diseases?
Yeah. Like, was the actual disease in them, or was it a mimic disease like we're doing?
It's in all cases we're mimicking it. So when we build a model, generally it's a transgenic model, which means we're putting the human mutant protein into the animal. So it's always made up to an extent. And then how well that animal recapitulates what you see in humans varies. This one's not that bad, really. There are far worse models, even than this mouse model here. I mean, it shows a lot of the biochemical changes that we think we see in humans. So we will benefit from the rat, but we're excited by the mouse. Yeah?
Two questions. How often do you have to do the intrathecal injections? Because I'm assuming obviously that the astrocytes are making more and more, with their DNA making more RNA that's sending off more proteins. And secondly, when you get rid of the protein, there had to be a point of this protein in the first place, although it's abnormal. What was the purpose of that protein originally if it were normal?
Right. First question first is about how frequently would the treatment be administered. That's something we don't know yet because we don't have our human drug made yet to know what the pharmacokinetics are, so to know how fast the body's going to break it down. There's a lot that's similar from drug to drug to drug, though, and the ASOs that we're producing now through research likely have an ability to be every 3 months. The SMA is delivered, I think it's more frequently than that, kind of in a loading phase, and then it backs off to every 4 months, I think. But again, that's a different chemistry because that's upregulating and we're downregulating. So I don't know what it will be, we don't know yet, but a good target for us would be an every 3 month delivery. And then the second question was, I've lost it already.
What's the downside of not having any protein at all?
Yeah, so that goes back to what we've talked about a little bit today is we talked about it last night, I'm not sure how much we talked about it today, understanding the role of GFAP just in normal wild type animals and in humans. And it's not so well described, but what we can do is knock it out completely in animals and see what happens. It's the advantage of looking at animals. And what we see generally, and this would probably be a much better question for Tracy or Albie, so I'll give a high level. Generally, we can knock it out completely and not suffer any side effects, which gives us confidence that we don't have a ceiling that we're pushed up against when we go to the clinic. There are some studies I know that show if you get a traumatic brain injury, if you induce traumatic brain injury in an animal that has no GFAP, it is harder for them to recover because we know this is an injury response. And as we think about developing a drug, we have to weigh that against the damage of continuing to have Alexander disease. Tracy or Albie, do you want to add more to that?
Yeah, so I would just say that, so there are a number of publications on mouse knockouts of GFAP, and we've made a rat knockout too that's not published yet, but the gist of it is that they look entirely normal, just from outward appearances. They're born in normal numbers, they grow up, they're running around the cage, they're hard to catch, they look fine. When you injure them in various ways, that's when you start to see some differences from control mice. Traumatic brain injury is one, so we could recommend avoiding traumatic brain injury, which is probably a good lesson for life. There's some evidence that they are more susceptible, or they suffer more severe outcomes following infectious or inflammatory diseases. But one of the problems is that they have such minimal phenotypes that there are very few people who've ever studied them. It's hard to get funding to work on them, so they're pretty much okay. And I think, as Anne summarized it, it's a matter of balancing, the risk. And our calculation is that this is worth the risk. I know.
Yeah.
So, when you get into, I don't know what the necessary, the technical steps are, phase 1, phase 2, clinical. Do you ever, how far, once you're in those, is it more procedural, or is there a chance that you can get to phase 1 and then not go to phase 2?
Yeah, so drugs fail at every stage of the way. Statistically, something like 10% of drugs that go into phase 1 succeed. Now, we like to think that we have better outcome than that, in part because all we're changing, we're working in, well, that kind of gross picture, we're working in a lot of different areas. We have something like 30 different clinical trials going on now in a variety of different areas, but in every case, it's a tiny little ASO. And it only differs by the sequence of nucleotides. So they tend to have similar properties from program to program to program. And we have a history of being able to predict based on sequences whether we're going to run into problems or not. So that gives us confidence. I think our success rate, once we get to the clinic, is probably going to be higher than industry average. But things can always happen. When we get into a toxicology program, we've selected the ASO we want to put in people, we put it in toxicology, something completely unexpected can happen, and then we're back to finding a different ASO to put into toxicology to get to the same place. So we can have setbacks.
So what stage is this in?
So this is still in preclinic. We need to have the actual ASO we want to put in people. So what we've been working with so far are ASOs that target the mouse gene, or that target that aren't necessarily the exact sequence we want to put in people. So once we have that, we can put it in animals and do those really standard box checking toxicology studies that the regulators require. And that takes time. That's at least 1.5 years of thorough safety investigation of the drug in animals before you go into people. And the idea there in toxicology is to be toxic. So you go to high doses so that you know what you're looking for when you get into people. You know what bad is, so you can look for that carefully in people. And then you can get the green light to go ahead and start clinical trials.
Thank you.
So we're still a few years away. Yeah?
With the Alexander patient population so small compared to other types of illnesses, it's obviously that's going to affect the FDA process. I think it's 3 phases generally.
Yeah, and
The third phase requires hundreds or thousands.
I don't want to steal Amy's thunder here. She's going to talk a lot about the stages of disease, or stages of development, but you're absolutely right. It's a different process to develop for rare disease. The burden is the same. You still have to demonstrate that the drug is safe and effective, but you can use creative ways to get there. Yeah.
Yes, I read somewhere that the FDA is in some kind of a fast-tracking process for the next generation of drugs, both including antisense. Are you aware of this? And would the antisense therapy be included in such fast-tracking process?
There are special programs with regulators both in the US and in Europe to get drugs to people sooner. And they are generally predicated on underserved population, which we are, serious illness, which we are, and great medical needs. So those options are available to us, things called fast track and accelerated approval and breakthrough status. These get you special favors with regulators where you get more interaction and better guidance so that when it comes to the trial, everybody's on the same page. So there are opportunities with regulators to make sure things move quickly. And yes, this kind of a treatment for this kind of population would be eligible for all of that. Yeah?
I was reading something that I think it was Laura Moody had done some studies with Dr. Essing about the GFAP in Alexander disease and in Rosenthal fibers actually degenerate faster than in normal. So that, would this therapy change that? And if it did, is there a way to test to see how much the dosage might change with every 3 months or 4 months? Is there a way to test how much GFAP is in there every time?
So I think I can answer that question. So what Laura's study did was look at the turnover rate of GFAP protein in brain. And we were actually expecting that the turnover rate would be slowed down because of problems with degradation. And instead, it was accelerated. And the turnover rate, which was the shift in population from one form to another, labeled in a specific way, was actually twice as fast in the mutant mice than it was in the wild-type mice. And that's actually really good news, and it really fits the antisense story that we published in explaining why 8 weeks after treatment, everything's gone. That we never expected it to work that well, and I think what happened is that as you start developing these accumulations of GFAP, part of that is because there's increased synthesis, and it takes a while for the cell to figure that out and to realize that it's got to start increasing its degradation. So eventually, in the adult animals, you reach a new balance, but it's a balance where you've already got lots more GFAP. But at that point, synthesis is matched to degradation. Then you come in with the antisense. You wipe out the synthesis part, but you're already primed for high-level degradation. And I think that's why 8 weeks was enough to just clear it all out. I think what will happen is, and we haven't done these studies yet, is that as you go through that recovery phase, it's going to re-equilibrate. And degradation is going to start to slow down again. And eventually, basically, through whatever dosing experiments we figure out how to do, we'll find an equilibrium point that's got a tolerable amount of GFAP. Maybe that's 0%, maybe it's 10%, maybe it's 25, I don't know. But basically at that point, synthesis will have matched degradation, and you're reasonably okay.
Going back to the FDA and the fast track, first of all, I have to say how thankful we are. It's truly amazing the progress you all have made. 20 years ago when we started this journey, there was almost nothing. We were often brain biopsy for a diagnosis, and it was just very different. So we're very thankful. However, talking about the fast track and accelerated progress, how do you get there? The only word that comes to me is like a special form of ending. That there is something on the horizon, and we need to wait. It's very, there's almost no words for that.
Yeah.
It feels helpless almost as a parent. So I guess that's two questions. Is there any, what do you do to get to fast track? And maybe that is the second question is, what can we do?
So in terms of regulatory strategy, that's something we'll work through which pieces of that make sense and to pursue and which don't. And I'm really very simple-minded about this. My goal and I'm on the development side, right? So my goal is how can we get to data as quickly as possible? We need data to demonstrate safety and effectiveness. We need enough of it. So we can talk with the agency about what's enough. But my sole purpose is to get to those data as quickly as possible. And everything else then kind of works its way out. What the trial design is, how many patients it is, how many trials it is, which of these sort of special regulatory paths we should pursue because they will speed things up, but which won't because they don't necessarily always speed things up. So that's sort of a thought exercise that we'll go through very soon to figure out. Our one goal is definitive data as quickly as possible. What's the best path to get there? Yeah.
One more, and then I think we'll move on. Right.
Yes, I agree with her. It's really a race against time. My daughter is already 26 years. And last year, Dr. Amy Waldman showed us her latest MRI. And it really shows a shrinking cerebellum. So, but there's hope.
Yeah. And I do, honestly, we know the need, we understand the need, and that's why I'm laser focused on that goal of getting to data as quickly as possible. So we know if this is an option. Yeah.
Okay, thank you. So